Welcome to the lecture on glycolysis. This is the first pathway that we'll study as part of the metabolism class, and it really does do a good job of going over some of the basic definitions we had in the introductory lecture. Again, the lecture objectives are going to be really important for knowing how much detail to focus on. There's a lot of detail here, and it's important, but there are things that are more important than others that you can spend your time on. Lecture objectives will help you with that. Okay, so glucose is the universal fuel for all human cells. To my knowledge, all human cells will use glucose as an energy source if they can get it. And the glycolysis pathway is going to be that pathway by which the glucose is utilized to generate energy, ATP, and electrons, reducing equivalents in the cell. So from this pathway, we generate ATP, and in this case, NADH is the reducing equivalent that we get by oxidizing the glucose. And we'll see substrate level phosphorylation for production of ATP. That's coming up. So overall, we're oxidizing glucose to pyruvate. Glucose is a 6-carbon molecule, and pyruvate is a 3-carbon molecule. It turns out one glucose will lead to 2 pyruvates being produced. But along the way, ATP is produced anaerobically. Now, if a cell has mitochondria, then the pyruvate that's produced by glycolysis can move into the mitochondrial matrix, and it can enter the TCA cycle, which will generate more reducing equivalents for the electron transport chain, and the electron transport chain then will support ATP synthesis by the ATP synthase, which is oxidative phosphorylation. And that, of course, is aerobic ATP production. Now, some cells are dependent on glucose as a fuel source for ATP production, and some of the easiest to remember are red blood cells. Red blood cells don't have mitochondria, so they can't do any aerobic ATP production at all. So they must have glucose. Glucose is the only source of fuel, and it's the only glycolysis is the only pathway for ATP production. But that's okay for what the red blood cell needs. It works out okay. Now, brain cells, on the other hand, have lots of oxygen, and there's lots of mitochondria in those cells, but the cell's blood-brain barrier really blocks a lot of materials from coming in. They let glucose in, but they don't let most fats in, for example, and so you don't get fatty acid oxidation. So here, brain is dependent on glucose because of the way the brain is designed. And we'll talk a little bit more about that as we go forward. Now, glycolysis will produce ATP anaerobically and are reducing equivalents as well. But there are other things that glycolysis can do. There's a side reaction, or what's called the pentose phosphate pathway, and that leads to a thing called NADPH, which is a lot like NADH, except for the P distinguishes this molecule, and it's going to be used in a couple of pathways that require electrons and reducing power. And then the other product is ribose 5-phosphate, which is going to be a very important starting material for the synthesis of nucleotides, nucleosides, and of course, nucleic acids. A lot of the amino acid biosynthesis, the ones we can make, the non-essential amino acids, we can make from glycolysis intermediates. And then when these amino acids are degraded, a lot of times they'll break down to glycolytic intermediates and then enter the glycolytic pathway here. Pyruvate is actually a precursor for fatty acid synthesis under an insulin signal, and insulin says we have way more glucose in the blood than we need, and it stimulates cells to take that glucose up, and the cells will take the glucose up and they very quickly meet their own energy needs. In tissues that synthesize fat, that insulin can tell them it's time to start making fat to build up membranes or to store fat or whatever. Glycerol 3-phosphate is going to be from the backbone of triglyceride synthesis. Glycerol 3-phosphate, we'll see, is right in the middle of the pathway. And then 1,3-bisphosphoglycerate, which is a really important molecule for this pathway for ATP generation. This is the molecule that can be converted to 2,3-bisphosphoglycerate. And we saw that molecule before in the hemoglobin lecture. That's the molecule that gets down on that little cavity in the hemoglobin tetramer, and it brings those 4 subunits together so that they cooperate more closely, and you get that sigmoidal cooperative oxygen binding. So here's the cycle overall, and I like these pictures because this is the summary picture, and it kind of gives you that overview feel of what it's all about. And if you can talk about the pathway in terms of this overall feel, then you're going to be way ahead on this stuff. So the glycolytic pathway, if you recall, is the oxidation of glucose to pyruvate, and ATP is going to be produced anaerobically, and so are reducing equivalents in the form of NADH. So there's the pathway, glucose to pyruvate. Along the way, the glucose molecule, which is very low energy, gets phosphorylated to glucose 6-phosphate. We've talked about that a couple of times. And so you raise the energy status of this molecule by phosphorylating it. And not just that, you isomerize this, you phosphorylate it again. Now you have a very super energetic molecule in fructose 1,6-bisphosphate. And this is the molecule now which will be oxidized directly to pull those electrons off, then generate ATP, and out comes pyruvate. Now what you'll notice is that 4 ATPs are produced during this phase right here, but you'll notice that 2 ATPs are going to be used by the kinases. And so you use 2, you get 4, you get a net 2 moles of ATP per mole of glucose coming out, and we'll talk about that coming up. So we say there are 2 parts then, as you saw. You use ATP early on, but then you get ATP after that. So the early phase is called the preparative phase. It's where you use ATP to raise the energy status of the glucose, 1 phosphorylation, 2 phosphorylations, to make this really interesting 6-carbon molecule that has 2 phosphates on it. It's a bisphosphate. And then you split that molecule into half, so you have 2 3-sugar phosphates. And those are the 2 molecules that you oxidize and use the phosphorus to make these ATPs. So preparative phase and ATP generating phase are the 2. Now if you remember, glucose is a 6-carbon molecule, and when you split it you have 2 3-carbon sugars, and in the end pyruvate is 3 carbons, and you'll have 2 of those. So the number of carbons will definitely mesh by stoichiometry. And if you like MCAT exams and you like these things, obviously summaries are going to be important, and the way the equations are balanced. You start with glucose and you get pyruvate. Glucose 6 carbons, pyruvate is 3 carbons, so 2 pyruvate for 1 glucose. That looks pretty good too. Now if you recall, you are generating reduced equivalents, in this case NADH. In fact, you get 1 NADH for each 3-sugar molecule, so if you get 2 pyruvates, you better have 2 NADHs. In order to have a reduced NADH, you better have an oxidized NAD+ that can receive those electrons. So that's why there's an oxidized NAD on the left, reduced NADH on the right. If you're going to be making ATP using ADP and some source of phosphate, you better bring in ADP and you better bring in phosphate. So when those things get hooked up together, you have the 2 ATPs are formed. And there are 4 protons that go along and 2 waters, and we'll see that that's part of the balancing of the equation. Overall, this pathway has a net negative delta G, which tells you that the pathway will be spontaneous as written. Given the enzymes along the way, as long as the enzymes are active, then each of these reactions will drive the pathway towards pyruvate. As we mentioned before, 2 ATPs are used in the preparative phase, and 4 ATPs are produced during the ATP generation phase, and so the net is going to be 2 ATPs. But if you have mitochondria and those pyruvate can enter the mitochondria, then you get the TCA cycle, electron transport chain, ATP synthesis by the ATP synthase, and you end up getting somewhere in the neighborhood of 25 moles of ATP per mole of glucose. So we will talk about stoichiometry a little bit more later on, but that just shows you the power of aerobic ATP generation. So here's the pathway overall, and I like to go through this just as a big picture. And if you've had biochemistry before, you probably had this pathway. You were probably asked to learn the enzymes and the molecules and the structure. I want you to understand the story of glycolysis. I want you to understand the logic of glycolysis. I want you to know how the cell makes decisions based on this reaction or that reaction, or who's going to be more and who's going to be less. And the way to do that is to have sort of a walking tour of the pathway. It's kind of like you're over at a friend's house and they say, let me take you out and show you my garden. And as you're walking through the garden, they're going to say, look over here on the left, I've got these really nice tomato plants. And right next to them, I've got these zucchini plants. And you get an idea of how the garden works by hearing the story of the garden. You may not know all the details, but you understand the story. So let's do that. So a little bit more detail. Here's glucose that comes in, and glucose is going to be phosphorylated at the 6th position by ATP, and the enzyme that does that for most cells is hexokinase. Hexokinase is the very high affinity version of the kinase enzyme. For liver and pancreas, we have glucokinase, which is the lower affinity form, and those tissues have a lower affinity enzyme like we've talked about before with a different glucose sensitivity. Once the glucose is phosphorylated, it's trapped inside the cell. Glucose can enter the cell as glucose, but it cannot exit the cell as glucose phosphate. Then it's important to isomerize this molecule. So you take the glucose, which is an aldose, and you just change that double bond, that carbonyl there, to the 2nd position. You create a ketose, in this case fructose. It's still phosphorylated at position 6. Another ATP is used using this enzyme phosphofructokinase 1. And phosphofructokinase 1 is designed to put a phosphate on carbon number 1 of this fructose molecule. And this is cool because now you have a phosphate on both ends of this 6-carbon molecule. The next enzyme that comes along is aldolase, and aldolase will split that fructose bisphosphate molecule into 2 3-carbon sugars here. And if you look at the structure, it's not identical, so you're going to get 2 different molecules. You're going to get dihydroxyacetone phosphate, because there's a phosphate on this end, and you're going to get glyceraldehyde 3-phosphate with the phosphate down here on position number 3. So it's hard to see the reaction. It's hard to know where the electrons are going right away. You can see that there's a double bond here. You see that ketone survives when this molecule is split, but where in the heck does this aldehyde come from? Well, we're not going to get so much into the organic chemistry. Sometimes we will, but here we're not. It's just more important to know the story. The story is fructose 1,6-bisphosphate is split in half by aldolase, and you get dihydroxyacetone phosphate, and you get glyceraldehyde 3-phosphate. It's the 2 products. Now, these are 2 3-carbon molecules, but only one of them can go forward. And in fact, it is glyceraldehyde 3-phosphate is the molecule that goes forward in the pathway. So this dihydroxyacetone phosphate molecule will be isomerized into glyceraldehyde 3-phosphate. And if you look, this is a ketose sugar, and this is an aldose sugar. And all you need to do is move that carbonyl carbon back to the end, and you move it back to the end right as you see there, okay? And if you flip it over, it ends up, the aldehyde ends up on that CH on that end, and then you have the phosphate on the carbon number 3. Okay, so now we have 2 3-carbon molecules. It only shows 1, but remember we have 2 of these, and this is where the first oxidation reaction takes place. As you see, you have oxidized NAD. It's oxidized. It doesn't have the hydride. And it will take a hydride off this molecule right there. That's the hydrogen that will come off. And you create that NADH. And when you create the NADH, you've now oxidized this molecule. But something else happens. Look at this. An inorganic phosphate that's present in the medium, just a PO4, is going to be used as a nucleophile, and it's going to bind onto that carbon here at the end. So you are now phosphorylating this molecule here on both ends. So you have the original phosphate and the new phosphate that goes on there. Again, the reaction chemistry is known. It's been described. You can look it up if you want. We're not really going to do the arrows and the electrons and stuff. Not so much. We're going to tell the story of them. But this is an extremely high energy molecule now. Look at this. You've got 2 very large phosphate groups attached to a very small carbon skeleton. And you've got another C double bond O here, and that oxygen is, of course, electronegative and has those electron clouds, and this OH. And so this is a very sterically hindered molecule, so there's lots of stored energy. It has a lot of free energy. And look what you do with 1,3-bisphosphoglycerate. You can take an ADP molecule, and you can take 1,3-bisphosphoglycerate, and this phosphoglycerate kinase enzyme will transfer the phosphate off of the 1,3-bisphosphoglycerate and stick it onto the ADP, and that will create an ATP molecule. And then you have a singly phosphorylated 3-carbon molecule here. And so that is called substrate level phosphorylation, where the substrate itself is going to be used to phosphorylate an ADP molecule to make ATP, and that's in the absence of molecular oxygen. So there's your anaerobic ATP reduction. And if you remember, there are 2 molecules of 1,3-bisphosphoglycerate, 2 molecules of 3-phosphoglycerate are produced, and 2 molecules of ATP are produced because of that. Now you have this enzyme called a phosphoglyceromutase. Again, you want to rearrange the structure of this molecule to set it up, and the rearrangement is going to be the phosphate going from the number 3 end to the number 2 position there. And you then do an enolase. An enolase is going to remove an H and an OH, and it's going to come from this H right there and this OH on the 3rd carbon, and you'll be left with a double bond there. And again, you've got a very sterically hindered, very constrained molecule with a double bond, a negative phosphate, and you've got that negative carboxyl group out there. So this molecule has a tremendous amount of free energy stored within its structure. And in fact, that's phosphoenolpyruvate is the molecule. And because it has so much energy, it can easily phosphorylate a molecule of ADP to create another molecule of ATP, and the product is going to be pyruvate. So again, 2 phosphoenolpyruvates are going to be used to phosphorylate 2 ADPs to give you 2 ATPs from this, and 2 pyruvates are formed. Okay, so you've got 2 ATPs and 2 ATPs, that's 4 total ATPs are produced, but of course you use 2 ATPs along the way to prepare this molecule, so the net ATP reduction is 2 net ATPs, and you get those 2 NADHs that we saw before. Now, one more thing to point out for you, the substrate level phosphorylation. In order for that to happen, you have to have a molecule, a substrate molecule that has more free energy than an ATP molecule. Phosphorylating an ADP is a positive delta G reaction, and you have to overcome that positive delta G in order to drive ADP and phosphate towards ATP. Well, if you remember in the what's high and what's low in the bioenergetics lecture, 1,3-bisphosphoglycerate and phosphoenolpyruvate both have more free energy than does ATP. And so those molecules, 1,3-bisphosphoglycerate and phosphoenolpyruvate, can both be used to make ATP in solution because they've got enough free energy to do that. Okay, so now we're going to look at some more detail here, a little bit of reaction stuff, but not quite so much. But this first committed step we saw before, the glucose is going to come into the cell, and when it comes in the cell, it's going to meet hexokinase, which is the enzyme that will phosphorylate that glucose using one ATP molecule. That's going to be for most tissues, but if you're a liver cell, glucokinase, or a pancreatic beta cell, for example, you have the glucokinase enzyme. And you make glucose 6-phosphate. Now, this is committed in the sense that the glucose is committed to the cell. It's now trapped inside the cell. But at this point, it's not committed to glycolysis, and that's because there are many things that glucose 6-phosphate can be used for in a cell. Glycolysis isn't the only reaction that would happen here. So glycolysis is one of these, but you could also do the pentose phosphate pathway, which we talked about, ribose 5-phosphate and NADPH. You can use this glucose 6-phosphate for glycogen synthesis, and there are other pathways like involving glycoproteins and glycolipids and stuff where you can use this. So it's kind of a common intermediate that you have, and the cell can make a decision based on what it needs. Here's a comparison of hexokinase and glucokinase, and I think we've seen this 3 times now at least, so we don't have to really go through this a whole lot. Hexokinase is going to be the enzyme that's found in most cells, like red blood cells, fast-twitch skeletal muscle, brain, where it's important that glucose come in and be phosphorylated immediately because glucose is the primary source of energy for those tissues. Those tissues are very serious about capturing that glucose. And then glucokinase is going to be more likely found in the liver and the pancreatic beta cells, and it has a lower Km, which means a lower affinity for glucose. So it's only going to be active when the glucose concentration is relatively high. Notice hexokinase is almost completely active, very, very quickly gains activity, even at the very lowest levels of substrate. And that just makes so much sense. I'm a red blood cell. I'm trying to find glucose. As soon as that glucose comes in the cell, I want to hit it with very high affinity, and I want to phosphorylate it to trap that glucose so I can get my energy. Okay, so the next committed step is going to be that phosphofructokinase 1 enzyme, or PFK1 is its nickname. And that's the one down here. That's where you take the Fructose, remember it's glucose 6-phosphate, is isomerized to fructose 6-phosphate, and that fructose 6-phosphate is going to be used by PFK1 to put a phosphate at position number 1 right here, okay? So when that happens, this is a committed step because you're committing another ATP to it, but when you make fructose 1,6-bisphosphate, there's really not a lot that molecule can do in a cell besides go forward to glycolysis. So at that point, that's called the committed step for glycolysis. And when it shows up, you know that you're on your way to making pyruvate. And because of that now, since this is the enzyme that's going to commit this molecule that has several uses in the cell, but PFK1 will commit it to glycolysis, you can imagine that PFK1 is going to be a very highly regulated enzyme. If the cell needs glycolysis and it needs the power of glucose oxidation, the cell will make sure that the PFK1 enzyme is turned on, that it's very active. If the cell has a lot of energy and it doesn't need to use glucose to make ATP, then the cell will work to shut down a PFK1 to make its activity lower. And there's a couple of molecular switches we'll talk about in just a few moments here. So it is considered to be thermodynamically irreversible because you're taking a mole of ATP and you're transferring that phosphate onto the number 1 position. Now fructose 1,6-bisphosphate has energy, it sure does, but it has less energy than the ATP molecule. That's why ATP has enough power to phosphorylate fructose 6-phosphate and to make that bisphosphate molecule. So if you go backwards, you have to do a positive delta G reaction. Of course, that's going to be tough to do. So thermodynamics are going to drive that one. It commits you to glycolysis because fructose 1,6-bisphosphate is a molecule that's destined for pyruvate. And of course, it becomes the rate limiting step of glucose. And what rate limitation means is, what is that one enzyme that's going to be the most targeted by regulation that either lets glycolysis move very quickly or one that would slow down glycolysis because of its activity? That's what we mean by rate limiting step here. And so after that, the fructose 1,6-bisphosphate is going to be cleaved into those two 3-carbon sugars. And if you recall, there is dihydroxyacetone phosphate and there is glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate is the one that goes forward, so you need to modify the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate for that. If PFK1, phosphofructokinase 1, is going to be that important rate limitation enzyme, we have to look at how it's regulated. What tells this enzyme to be more active to let more glucose oxidation take place, or what tells this enzyme to be less active so you have less glucose oxidation going on? And that's because there are a couple of very powerful allosteric modulators for this enzyme. And one of them is going to be AMP, which is the lowest phosphorylated adenosine thing we have. Okay, so there's ATP, ADP, and AMP. AMP is going to be a very strong positive allosteric modulator, which will activate this enzyme and make it more active. And then ATP is a very strong negative allosteric modulator of PFK1, ATP. So why would that be? Okay, so if you look over here, you see the normal enzyme, and you see it has a sigmoidal shape, would let you know that PFK1 is an allosteric enzyme, it's cooperative. And that's just going to be under normal conditions. And so when fructose 6-phosphate is present, if you have a little bit of substrate, you don't have much activity. But as the fructose 1,6-phosphate binds, you're going to get those cooperative conformational changes. The active sites will open up, the enzyme will become more and more active, and you see a very rapid rise in activity up to Vmax. When AMP binds, because it's a positive allosteric modulator or an activator molecule, it's going to help uncouple those enzymes from each other so that each enzyme can bind substrate and each enzyme can become active more quickly. And you'll notice in the presence of AMP that you lose that sigmoidal shape of a cooperative enzyme and you get more of a hyperbolic or a Michaelis-Menten. So AMP is so powerful it almost completely removes the cooperative nature of PFK1, and you see it even very low levels of fructose 6-phosphate. You get very high levels of enzyme activity. Now there's another molecule there, fructose 2,6-bisphosphate. We'll hold off on that one for a while. That's how you stimulate when you're thinking about in the liver. So we'll get there. So this is really good for muscle. If you think about a skeletal muscle and the muscle is running or exercising, a lot of exertion, a lot of ATP use, and so the ATP level in the cell is going to go down, down, down because the ATP is getting used up. And that's going to cause the AMP level in the cell to get higher and higher and higher. And so as that muscle cell is getting lower and lower ATP levels because it's using the ATP, as the AMP starts to rise, the AMP is a positive allosteric activator for this enzyme. It binds to those activator sites. It turns PFK1 on. It makes the enzyme much, much more active, and that helps glycolysis produce ATP faster, which is exactly what the muscle cell wants. The muscle cell wants more ATP, and AMP levels signal it's time to make more. What about ATP then? If ATP really is one of the products of the glycolysis pathway, if your cell has lots and lots of ATP, does your cell want to use more glucose to make more ATP? Well, of course not. If ATP levels are high, you don't want to use that precious glucose to make more ATP. You want to use the glucose to do something else. And so what we find out for ATP is when the level of ATP is kind of low, you see a maximum activity. But when the ATP level gets higher and higher, you see that the enzyme activity level will drop off a lot. So one of the questions you may have is, why does it rise at low ATP? And then why does it come down somewhere around 4 or 5 millimolar ATP? Well, if you recall, this is a kinase. PFK1 is a kinase enzyme, and it needs ATP as a substrate to be used to phosphorylate the molecule. And so just like any other enzyme, if you give it more and more substrate, you get an increase in velocity. And that's what this is showing you here. But ATP in this case is also a negative allosteric modulator. And as the ATP gets higher and higher and higher, it binds those allosteric binding sites. And because it's negative and inhibitory, it turns that enzyme off. And you see very, very rapidly. Now, when muscle cells are at rest, they typically have 4 to 5 millimolar of ATP just in the muscle cell, which is considered a pretty full amount of ATP. And you see under those conditions, this PFK1 enzyme is almost completely shut down. But if the muscle starts to move and you start to get activity and these things are going on again, then very quickly AMP levels start to rise and you're going to get a shift to the left as you expect with the AMP. And you see that in the presence of AMP, you can completely override that ATP inhibitory effect. That's what that line is showing you there. So even if the muscle cell has a lot of ATP, if AMP starts showing up, it's going to have a stronger influence and get this enzyme moving. And again, we'll talk about fructose 2,6-bisphosphate a little bit later. And of course, there's a great clinical significance here. If you think about glycolysis in skeletal muscle, if a person has trouble with enzymes in their glycolytic pathway and they can't produce ATP in tissues where they really need to make ATP from glucose, then they're going to have a problem. So for example, in red blood cells, if there's a deficiency with the glycolytic pathway, the red blood cells can't use glycolysis to make their ATP, and that's going to cause the cell quite a bit of harm. Or if you're in skeletal muscle and the muscle's exercising and exerting and you can't regenerate that ATP, it's going to lead to muscle weakness and muscle cramps because it just doesn't have enough energy. And the enzymes that tend to show up in deficiencies are going to be PFK1, which is the rate-limiting enzyme for glycolysis, but also the enzyme at the very end. Pyruvate kinase is another enzyme that shows up at the deficiency, especially in skeletal muscle, where people have, when they start to exercise and they have a lot of pain, and it's obvious there's something wrong with the energy production in their muscles. Okay, so let's go back now after we have the fructose 1,6-bisphosphate, and that's the molecule that gets split by the enzyme aldolase. And aldolase makes those 2 3-carbon molecules. Now what's interesting is there are 3 isoforms for aldolase, and this is a little bit confusing, and it's going to be important later on for fructose metabolism and for gluconeogenesis. There are 3 different forms of aldolase, A, B, and C. Aldolase A is going to be found in developing embryos, during pregnancy there. But it's also found in adult skeletal muscle. So fast twitch skeletal muscle will have aldolase A, and of course red blood cells will have this as well. Aldolase B is found primarily in the liver. And that aldolase B is going to be an enzyme that's involved in fructose metabolism. We'll see it's a couple of lectures away now, but when a person is fructose intolerant and they can't digest fructose, a lot of times it's because they don't have the aldolase B enzyme. Now that's not a problem because aldolase B is in the liver that's involved in fructose metabolism, and then aldolase A is going to be the one in skeletal muscle and red blood cells that help them do that. And then, of course, alpha C is going to be in brain and nervous tissue. Okay, so if you have an A deficiency, which is true in red blood cells, that tends to bring your myopathies and your hemolytic anemia here for muscle myopathy and red blood cells hemolytic anemia. And then alpha B is going to be for the fructose metabolism. Okay, as we move along now, glyceraldehyde 3-phosphate. When you extract the hydride, you take the hydrogen, the proton, and you take the 2 electrons. That's the hydride group. And so you're taking more electrons than just one single atom, and that's going to lead to an oxidation. The electron goes to NAD, so NAD becomes NADH. The electrons are there, and so now NADH is reduced, and the molecule 1,3-bisphosphoglycerate is going to be oxidized because it's lost electrons. And then you see substrate level phosphorylation, where the 1,3-bisphosphoglycerate is going to give away a phosphate to ADP to make ATP with that 3-phosphoglycerate. And if you keep going again, there's just that rearrangement of structure to set this thing up. You remove water from right there, and that's going to give you a double bond right next to that phosphate and the carboxyl group, and that gives you phosphoryl pyruvate. That phosphate now has super high energy, so it very easily will be transferred onto the ADP to make ATP by the enzyme pyruvate kinase. So pyruvate kinase is that enzyme a couple slides ago that shows up especially in skeletal muscle. If it's deficient or has lower activity, that's why the skeletal muscle can't get enough energy to do the contraction it needs to do. So when we get to pyruvate, we're at the end of the glycolytic pathway. That's the end. Pyruvate is the product and you're done. Pyruvate, NADH, and ATP are all produced here. So what happens to pyruvate? Well, pyruvate has 4 fates. And a fate just means outcome. What's going to happen to the pyruvate that's produced? And there are 4 things that can happen. If you're a cell that has mitochondria, then that pyruvate can move into the mitochondrial matrix. It can participate in the TCA cycle. And the normal route for pyruvate to enter the TCA cycle is to go to acetyl-CoA, and acetyl-CoA is the molecule that gets used in the first step of the TCA cycle. But there's another way to go. We saw that in the metabolism introduction lecture. We had 2 pathways, and that would be pyruvate going to oxaloacetate. So sometimes you'll have a lot of acetyl-CoA and the TCA cycle says, wait a minute, I don't need more acetyl-CoA. I've got lots already. Can you help me out? And pyruvate says, yes, I sure can. I can go over here and make more oxaloacetate because it's these 2 molecules that come together in the first step of the TCA cycle to give you citrate. Now, the other things that you can do with this, if you don't have mitochondria, or if you have very limited mitochondria capacity, or if you have no molecular oxygen, so the mitochondria isn't really working properly, pyruvate can be turned into lactate. And if you see this here, it's called a reduction. Pyruvate is more oxidized, lactate is more reduced, because you're going to be investing electrons back in that pyruvate molecule to make the lactate. And then since you can't use the lactate, it becomes just a product the cell doesn't need, so it will eject it out into the blood. And it will circulate in the blood until it comes across a tissue that can use that lactate. And as you can imagine, any tissue that has a high level of mitochondria with lots of oxygen, they can use lactate very easily. They bring it back in the cell, they convert it back to pyruvate, and they send that pyruvate right into the TCA cycle so they can get all that energy. A good example of that is cardiac muscle. And the last thing you can do is you can transaminate. Pyruvate is a 3-carbon molecule that looks an awful like alanine, except alanine has 2 carbons and an amine group. And so transamination is a way to switch an amine group on and off. And we'll see that coming up. There'll be a whole bunch more lectures from now, but transamination is good. But this is the reaction where you go from alanine to pyruvate when you're going to be synthesizing glucose. The starting material is an amino acid. You turn it into a carbon skeleton, then you can send it back up towards glucose. Okay, so what happens with that NADH that's made inside the cell? Okay, if you have mitochondria and the mitochondria is working, then the TCA cycle lives inside the mitochondrial matrix. The electron transport chain is in the inner mitochondrial membrane, and the ATP synthase is on the inner mitochondrial membrane as well. So we have to figure out some way to get those electrons to the electron transport chain so they can lead to ATP production. So if you may or may not, I don't know if we talked about this yet, but there is no transporter for the NAD or NADH to get across that membrane. Okay, it's just not possible. It's just not there. I don't know why. I can never figure that out. So we have to figure out some way to get those electrons across the membrane. How is that going to happen? Well, we use the thing called the glycerol 3-phosphate shuttle or the malate aspartate shuttle system. And the way this works is here's the glycolytic pathway. There's glucose, there's pyruvate. So they're just showing you here in one step, this is the glycolytic pathway we saw before. And it's showing you that 2 net ATPs are being made. It's showing you that 2 NADHs are being made. And we know now that the ATP can go and be used by the cell for whatever it needs for its energy. But then the NADH, we're trying to get those electrons to the inside of the mitochondrial matrix so those electrons can make it to the electron transport chain. And there is no transporter for NADH or NAD+ on this intermitochondrial membrane. Well, nature helped us out a little bit here, and there's this thing called the glycerol 3-phosphate shuttle or the malate aspartate shuttle, and you just take the electrons that are held on NADH, that hydride, and you give that molecule, those electrons to some of the molecule here. You've got some molecule which is more oxidized. You've got NADH, which is reduced. You hand the electrons off to the oxidized molecule, it becomes reduced, and at the same time, the NADH that used to be reduced is now going to be oxidized. And that's good for two reasons. You can give this NADH, you can give those electrons to a molecule that has the ability to get to the intermitochondrial membrane and hand the electrons off. So you found a molecule that can do something that NADH can't do. And the other thing that's good for you is it's going to regenerate this oxidized NAD because you remember for glycolysis, one of those reactions near the middle of the cycle had electrons coming off of glyceraldehyde 3-phosphate, and they were given to NAD. So you need a source of NAD to accept those electrons, and this allows it. Okay, let's start with the glycerol 3-phosphate shuttle because it's a little bit easier to understand. So here's glucose, and it goes to pyruvate, so there's our pathway, and we know that during that pathway, that NAD, which is oxidized, is going to take electrons away from the, in this case, glyceraldehyde 3-phosphate molecule, and those electrons are given to the NAD to make NADH. NADH is reduced, it has the electrons now, but it can't cross the intermitochondrial membrane. The partner molecule that it will interact with is going to be dihydroxyacetone phosphate. Do you remember dihydroxyacetone phosphate? When the fructose 1,6-bisphosphate molecule is produced during glycolysis, the aldolase enzyme will split that 6-carbon molecule into 2 3-carbon molecules. One of them is dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. So dihydroxyacetone phosphate is a product molecule, and I mentioned that it gets converted over to glyceraldehyde 3-phosphate so the pathway can go forward. But not all of the dihydroxyacetone phosphate will go forward. There's a pool, which means the supply of that molecule. There's pools of all these intermediates, and the DHAP, dihydroxyacetone phosphate, is just one of those pools. And so if you have a cell and you need to get these electrons in, you just grab one of those DHAPs and you give it the electrons from the NADH. And when you reduce dihydroxyacetone phosphate, you get glycerol 3-phosphate. Glyceraldehyde 3-phosphate is more oxidized. Glycerol is more reduced. So glycerol 3-phosphate is not the same as glyceraldehyde 3-phosphate in glycolysis. But it has the electrons, and that's good. And it turns out that glycerol 3-phosphate has the ability to get here to the surface of the mitochondrial membrane and give these electrons off to a molecule of FAD, flavin adenine dinucleotide. And the FAD is oxidized. When the glycerol 3-phosphate comes down, they bind together, the electrons come off of glycerol 3-phosphate and they go on to FAD, and that produces FADH2, which is the reduced form of the FADH2. And those electrons now are sitting in the electron transport chain, and it will give those electrons off to the chain. And we'll talk about that coming up in a couple more lectures here. So meanwhile, when you took the electrons off of the NADH, you gave those electrons to hydroxyacetone phosphate, you created glycerol 3-phosphate, and in the process you reoxidize the NADH to NAD+. Now that NAD+ is ready to go over here and pick up some more electrons from the next molecule that is being oxidized on its way to pyruvate. So it works together really, really well. Now, the idea is similar for the malate aspartate shuttle. It's just a little bit more complicated, and it's a little bit hard. If you've had biochemistry before, you've seen the TCA cycle, and you know about the relationship between malate and oxaloacetate. If you haven't done biochemistry before, you don't remember the TCA cycle, you'll have to hold on until the next lecture where we see how this works together. But part of the TCA cycle is to take malate and convert it to oxaloacetate. This is a TCA cycle reaction. And one of the things that this TCA cycle reaction does is it generates NADH. And that NADH is going to go to the electron transport chain. This is all inside the mitochondrial matrix. That's how the TCA cycle works, and that's how you feed electrons to the electron transport chain. Well, it turns out malate and a form of oxaloacetate have the ability to go through a transporter. See, malate can go through the intermitochondrial membrane. It can get converted to oxaloacetate, which can be turned into aspartate by a transamination reaction. That's coming up in a couple more lectures, by the way. And aspartate can leave the mitochondria. It can go out through that intermitochondrial membrane. And as soon as it gets outside, it gets turned right back into oxaloacetate. And the oxaloacetate can be reduced back to malate by taking electrons from an NADH. Those electrons go on to oxaloacetate to help make malate. Then malate can come back in through its transporter. Okay, and that's going to be very good because glucose to pyruvate. Glucose, they're going to be producing NADH, and you need to get those electrons into the mitochondrial matrix. Now, we haven't talked much about transamination, but I'll just give you a little foreview here of what it is. For transamination, you'll have an amino acid, and that amino acid will have an amino group on the alpha carbon. You remember that. But then you have what's called an alpha keto acid, where you have a ketone right next to a carboxylic acid. And alpha means the first position, so alpha keto acid. And you'll notice that there is no amino group here. So the alpha keto acid and the amino acid can react together, where you're going to just switch the carbonyl C double bond O for the amine group. And look what happens. Oxaloacetate becomes aspartate. You lose the C double bond O, but you pick up that amine. And over here, a glutamate that had the amine is going to lose the amine group, and it's going to pick up the C double bond O. Pretty complicated chemistry, how it works, and lots of steps involved in this. But these are called transamination partners. You will always see oxaloacetate becomes aspartate, and aspartate becomes oxaloacetate. And you will always see glutamate, and you'll see alpha ketoglutarate working together. Now, that's not to say that they're always the transamination pairs, but pretty commonly here. And that's why you can see that oxaloacetate can be converted to aspartate just because it has a transporter that takes it out. And glutamate over here is going to be involved, which is why we see glutamate coming in. But the point is that there are electrons out in the cytoplasm, and by handing those electrons off to a molecule that has a transporter, you can transport those electrons into the mitochondria, where they can be reconverted to that carrier, and that carrier can then carry them to the electron transport chain. Okay, so what happens when you don't have mitochondria, or there's not enough oxygen to support mitochondrial ATP synthesis or something like that? Under those conditions, there's nothing you can do with pyruvate, and it becomes the end product, and it's kind of like the trash. The cell needs to put the trash out. But the cell has a little problem here. It says, for glycolysis, I know I need to extract some electrons, and I need to put those electrons on an NAD because I need to make an NADH. And so I better have a supply of NAD so I can give it some electrons. And so the cells figured out a long time ago that pyruvate and lactate are oxidation reduction pairs. And if pyruvate starts to build up because the cell can't do anything with it, it can take advantage of the NADH that's produced, and it can transfer that hydride onto that carbonyl carbon, and you can reduce that carbonyl down to an alcohol, and you make this molecule lactate. And lactate can be released out of the cell and into the blood just like you'd put out the trash. That's now gone. You've gotten rid of your pyruvate, but in the process of turning it into lactate, you have regenerated your NAD, which allows glycolysis to keep going. Now, this would be the case if there's no mitochondria, or if your oxygen level is too low, or maybe you don't have enough capacity of mitochondria to really turn all of the pyruvate into oxidative phosphorylation. So for example, red blood cells and certain in lens of the eyes and fast twitch skeletal muscles, production of lactate is going to be pretty important. So red blood cells can only produce lactate, and skeletal muscles, they'll produce some lactate and they'll put it out. They have some mitochondria, but not a big amount. So if you're just sitting around resting or walking or have some light activity, your skeletal muscle mitochondria can keep up very nicely. But when you're under a lot of exertion, when you're exercising or running or a lot of intense activity, you're using glucose and glycolysis much faster than the mitochondria can keep up with. And so that pyruvate gets turned into lactate and ejected from the cell. Okay, so the fate of lactate, remember the word fate just simply means what happens to it. Where does it go? What does it become? And so it's going to be taken up by tissues that have excess mitochondrial capacity. Any tissue that can take a molecule and then oxidize it. And that's going to be cardiac muscle primarily. And skeletal muscle too. There's slow twitch skeletal muscle, which is like cardiac muscle. It has lots and lots of mitochondria. It likes fat. It likes lactate. It likes things that it can use for oxidative ATP production. And then, of course, liver will take lactate up during times of fasting when liver needs to make glucose to maintain the blood glucose level 80 to 100 milligrams per deciliter. And one of the precursors it can use is lactate. Lactate gets turned into pyruvate, and then pyruvate goes back to glucose. So that's a really nice energy source for liver during fasting. And there it is, the Cori cycle, which is showing you how this works. The red blood cell, for example, glucose goes to pyruvate. The cell can't do anything with the pyruvate. It says, I'm stuck. And so it turns that pyruvate into lactate so it can regenerate its NAD needed for glycolysis. And the lactate then gets ejected. And the liver says, oh boy, I can take this. I like lactate. It uses lactate to synthesize glucose. And there you have it there. That's called the Cori cycle. We'll see a couple cycles like that. One tissue's trash is another tissue's treasure. And that's so true in metabolism. And like any other pathway, the cell is going to say, if I have glucose and I can use glucose and I can do glycolysis, what do I want to do with that pathway? And the cell will say, if I need energy, if I'm running low on ATP, you better bet I'm going to make my glycolytic pathway as active as it can be because I really want to have a lot of ATP. And so you'll see enzymes now, these green things are going to be allosteric activators. So you remember AMP is a strong allosteric activator saying, oh, AMP levels are high. That means there's not a lot of ATP. We better do glycolysis so we can make more ATP. And so you stimulate the fructose 1,6-bisphosphate, and you the PFK1 enzyme to make that bisphosphate, and then down here pyruvate kinase is going to stimulate that ATP production as well. But of course, if your ATP levels are high, the cell's going to say, wait a minute, my ATP levels are really high. I don't need to be making a whole lot more ATP. And this is where you see that ATP will shut down that PFK1 enzyme like we saw before. And very similarly, ATP will shut down the pyruvate kinase. Remember the logic of the cell. If ATP is high, you do not need to make more ATP. But if ATP is low, you better make more ATP. And so you'll have these allosteric modulators that will turn a pathway on or turn a pathway off, depending on what it's producing. So this brings us to the last big concept, and that's the idea of adenylate charge, or the energy content of the cell. And adenylate charge is kind of, I don't know, you see it in some books and sometimes it's just called the energy level. But what this is it's showing you how much ATP you have in a cell compared to the total adenine forms that you have, AMP, ADP, and ATP. So the amount of ATP that you have available on hand to do that work of the cell is going to be a ratio of all these different forms. Now you can imagine if your ATP level is very high, very big, you wouldn't expect to have a lot of ADP, you wouldn't expect to have a lot of AMP, you expect those to be small, and if you have one number over the other, it's going to be almost 1. But if your ATP levels are small, then you'd expect ADP and AMP to be big, and you have a small number divided by a big number, that's going to give you a value less than 1, and so your adenylate charge is going to be lower than 1, quite a bit lower than 1. So you can imagine if you have a catabolic pathways are designed to make ATP. And so when your adenylate charge is low, you better think the catabolic pathways are going to be on very active. And if your adenylate charge is high, then you don't need to be making ATP, and so catabolic pathways are inhibited when the energy charge is high, when you have a lot of ATP. Likewise, your anabolic pathways, now they require energy. They want to use energy. And if you have a high energy charge, you have a high adenylate charge, that's going to turn on your anabolic pathways. There's a lot of energy. Let's use that energy to synthesize, build, and grow. So this can be demonstrated pretty nicely by looking at the amount of ATP, ADP, and AMP that's present in, say, a skeletal muscle during times of rest versus times of exercise. During exercise, the muscle's working hard, it's using a lot of ATP, it's very energetically, doing a lot of work and very active here. So it would make sense when you're at rest, your cell has all the stuff it needs, it's not really working very hard, it has time to build everything up, so your ATP level is high. But as soon as you start exercising, you use that ATP to do the work of the contraction and stuff, and so the ATP level is lower. And if you're going to use ATP, the first product you make is ADP. And so if exercise causes your ATP level to go down, it would make sense that exercise makes your ADP level go up. The green is bigger because you're exercising. Well, that's fine now, but ADP is not ATP, and your cell needs ATP to keep exercising and keep exerting. There's another enzyme called adenylate kinase. The adenylate kinase takes two molecules of ADP, it pulls the phosphate off of one ADP, and it sticks it on the other ADP. And so one ADP becomes an AMP, a monophosphate, and that takes the other ADP, you add a phosphate, it becomes an ATP. And because of that, at any given time, the amount of ADP you have is going to be equal, half of that can be converted to ATP and half can go to AMP. So when we look at our adenylate charge equation down here, we see ATP divided by ATP, ADP, and AMP, but you'll notice there's 1/2 ADP sitting there. And that tells you at any given time, half of the ADP can be converted to ATP because of adenylate kinase. Now, how does this work? You see that ATP, the adenylate charge, is going to be the ATP divided by the sum of all three concentrations. And so ATP is high. It doesn't change that much with exercise, maybe about 20%. This one doesn't change a whole lot with exercise, maybe about 20%. But look at AMP. AMP is going to be the big secret here. When you're at rest, the AMP level is actually very low. And so the number in this denominator is pretty small. It doesn't really have a big effect on the fraction. But during times of exercise, AMP is several-fold bigger than it is when you're at rest. And so the AMP number is now very large compared to what it was before. And because it's in the denominator, the energy charge number can become quite a bit smaller. And so that shows you how AMP becomes such a powerful signal for the need for energy. Now, of course, cells aren't reading these graphs and cells don't get little telegraph messages that the AMP level is high. The AMP molecule is going to be that positive allosteric modulator that will stimulate, in this case, the glycolytic pathway to turn glycolysis on to produce more ATP more quickly. Okay, so here's a question. Let's look at this. During vigorous exercise, the adenylate charge in a skeletal muscle cell will decrease from 0.85 to 0.75. Which of the following is most likely responsible for this change in adenylate cyclase? When you see a question like this, you think, well, wait a minute, what is the adenylate charge? Skeletal muscle. Adenylate charge tells me how much ATP I have compared to ATP, ADP, and AMP altogether. And I know when the number is higher, it means there's more ATP. When the number is lower, there's less AMP. So what is it about skeletal muscle and exercise that causes the adenylate charge to go down? And you say, yes, I remember when skeletal muscle exercises, AMP levels start to increase, and that AMP becomes a very powerful signal that it's time to make more ATP to give more energy to the muscle so it can do the work it needs to do. So clearly, AMP must be the most important signal that tells the energy charge what's going on, adenylate charge. So if you were looking at answer C, you would be absolutely correct. Yep. And AMP, of course, is going to activate that PFK1. And we haven't done glycogen yet, but we're going to find out that glycogen breakdown is stimulated by AMP as well. So glycogen produces more glucose, and glycolysis will oxidize more glucose under an AMP signal. So here's a nice question, and the question's a little bit ahead of where we are right now, but I still want to have it in there so you'll understand the fate of the electrons, but you may not know about the stoichiometry numbers yet. But just, we'll keep a post-it note on this because this is the right way to ask this question. I'm a little ahead of myself on this, but it's okay. So here's the glycerol 3-phosphate dihydroxyacetone phosphate shuttle system that carries electrons on NADH to the intermitochondrial membrane. And they're asking you, what is the fate of the electrons that are carried by this shuttle? So what the question is getting at is glycolysis produces NADH, but NADH cannot get across the intermitochondrial membrane to carry the electrons to the inside, to the matrix. And that's where they need to be to go to the electron transport chain. And if you're thinking about this, you're thinking, oh, I remember that, the glycerol 3 shuttle system. The NADH reduces DHAP, and that makes glycerol 3-phosphate. Glycerol 3-phosphate can get in there right at the surface of the mitochondrial membrane. It can bind to a protein and give the electrons away, and that protein just happens to give them directly to the electron transport chain. So fate means what happens? Where does it go? Where does it become? Where does it end up? What is the fate of those electrons? And the answer is they're going to be given to a flavoprotein, and that's going to lead to the formation of ATP, okay? And it turns out to be 1.5 moles of ATP. And we'll see this when we do the electron transport chain, which is in just a couple more lectures. And the story is here. This is where we are right now. Glucose to pyruvate, there's your NADH that's being produced. Dihydroxyacetone phosphate is more oxidized. Glycerol 3-phosphate is more reduced. How do you know that? You know that because NADH is the reduced form of NADH, and it's the thing that's binding to dihydroxyacetone phosphate. So if NADH is reduced, dihydroxyacetone phosphate is going to be more oxidized. And so that dihydroxyacetone phosphate will pick up those electrons. It will become reduced. It will become glycerol 3-phosphate. And that will regenerate your NAD to go back to glycolysis. Now that glycerol 3-phosphate can easily find its way into the membrane here, to the intermitochondrial membrane. It binds to a flavin protein, and that flavin is oxidized. But when glycerol 3-phosphate binds, the electrons jump onto the FAD to reduce it. You get that FADH2 molecule. The electrons are now on that FADH2, and glycerol 3-phosphate is reoxidized back to DHAP, and it can just go back to whatever it was doing before. And then you see here those electrons are given directly to the electron transport chain, and we will see why it's 1.5 ATPs per NADH in a couple of lectures coming up. In the absence of mitochondria or molecular oxygen, what happens to the pyruvate generated by glycolysis? Okay, so pyruvate is the end of the glycolytic pathway. It's the last molecule formed. If you have mitochondria, you know the pyruvate goes into the mitochondrial matrix and it enters the TCA cycle, and that feeds the electron transport chain and aerobic ATP synthesis, right? Okay, but what happens if you don't have the mitochondria? Pyruvate is just trash. But is the pyruvate going to be kicked out of the cell? Well, if you remember, the cell that doesn't have mitochondria needs to regenerate its oxidized NAD, and the way you do that is you take pyruvate and you'll take the NADH produced by glycolysis and you move that hydride onto pyruvate to create lactate. And when you create lactate, that turns the NAD back to NAD+, or NADH to NAD+, and that allows glycolysis to go forward. So you're looking for an answer that has something to do with it's converted to lactate. Do you see that here somewhere? Yeah. It's reduced to lactate, generating 1 NAD. That's the right answer. And this is wrong. It's not oxidized to lactate, generating NADH. NADH is what's produced by glycolysis that's used to reduce pyruvate to lactate. So yeah, there you go. Answer C. Alrighty. And there's the reaction. There's lactate dehydrogenase that does that reaction.